There Might Be Hope, Chapter 2, Faith Until Sight. Since the 1950s, USDA scientists have led the world in researching soil's physical attributes, textural and mineral composition, the effects of vegetative cover, weather, tillage, and slope on erosion rates have all been meticulously studied. However, our vast knowledge of soil's physical characteristics and mechanisms never fully answered the question of what causes farmland erosion or how to stop it. As science advanced through the 19th century, two dueling views battled for soil science predominance. Were soils primarily chemical or primarily biological entities? Chemical advocates believed that soils were primarily lifeless mineral mediums for plant growth that chemicals passed through on their way to root systems. Biological advocates believed that the key to soil fertility would one day be found through managing and balancing soil microbes and nourishing beneficial microbial communities. The problem was, in the 1800s, they couldn't find these communities. 19th century laboratory scientists' best efforts to locate and cultivate soil microbes on agar plates and in petri dishes generally proved unimpressive at best and entirely fruitless at worst. This led many to the belief that lack of growth on this medium was sufficient to justify the conclusion that the organism did not exist. All efforts to isolate the specific organisms concerned in the process failed. Farmers and microbiologists often intuitively felt that there was more to their soils, especially healthy ones, but evidence was not on their side. Just prior to World War I, German scientist and future Nazi chemical warfare specialist Fritz Haber took Carl Bosch's small-scale lab methods of extracting nitrogen from the atmosphere, and with the aid of substantial natural gas inputs, replicated the process on an industrial scale. Known as the Haber-Bosch process, its immediate product was ammonia, but the ultimate goal was massive ammunitions production. Decades later, following World War II, rather than be torn down, former munitions factories remarketed their primary product, ammonia, as synthetic inorganic fertilizer. Prior to this, the ability to selectively add massive quantities of an isolated chemical element to our crops had simply been out of reach. Just in time to facilitate the spread and distribution of these new fertilizers came the advent of powerful hydraulic tractors and farm equipment. The spark to this oxygen enkindling was Norman Borlaug's revolutionary hybridized seed creations. Immigrating from Mexico to the U.S. and thereafter sweeping around the world, the trio led to exponential increases in agricultural crop production. Agricultural yields exploded. For almost a century, since the USDA began tracking crop yields, the average U.S. cornfield had steadily yielded around 25 bushels an acre. From 1950 to 1959, that average grew to 44 bushels an acre, and then 70 in 1960. By the 1990s, cornfields were doling out 123 bushels an acre. Gains in potatoes were even more dramatic. Yields more than tripled from the previous national average of 5,000 pounds of potatoes per acre to 16,500 pounds in the 1950s. And 40 years later, by the 90s, that figure more than doubled to 33,000 pounds per acre. In a short 50 years, American fields went from producing an average of 5,000 pounds of potatoes to over 33,000 pounds of potatoes per acre. Gains in other crops, though perhaps not as dramatic, were substantial. The Green Revolution had definitely arrived. It saved millions from starvation, and proved that the purely chemical approach to the soil brought results. There was tangible evidence that purely chemical soil manipulation worked. Here was something soil microbiologists lacked. Alongside these technological advancements, evolutions in finance, along with increasing land prices and equipment costs, birthed the consolidation and specialization of formerly diversified operations on an increasingly massive scale. Survival for many agriculturists meant taking a page from the assembly line. Small-scale family farmers, lacking the volume of land necessary to produce competitive volumes, began falling by the wayside. Survival in an already risky endeavor virtually mandated large acreage, monoculture, or single-crop farming. Farms increasingly looked to new government crop insurance and subsidies as potential bailouts to assuage the additional risk and debt they assumed in transitioning from traditional diversified operations to potentially more lucrative monoculture operations. With this increasing specialization of agricultural operations, the traditional animal and plant agricultural cycle that had recycled local manure by applying it to crops disappeared. Livestock production began to also be consolidated into confined animal feeding operations, or CAFOs, where livestock waste that had formerly been an essential crop nutrient was, likely for the first time in human history, more problem than blessing. By the 1950s, the inherent difficulties and often fruitlessness of soil microbiology research combined with the emergence of patentable and hence far more lucrative means of addressing soil fertility gave root to the idea that soil microbes were probably of little value and their utter extermination a boon. The protozoa were looked upon as merely injurious forms or as the enemies. The fungi were considered either nuisances or dust contaminants. None of these organisms, however, were given sufficient consideration in any systemic study of the soil population, if they were considered at all. Selman Waxman, Nobel Prize winner. Somewhere along the way, these presuppositions began to be taught as established and then static truth to generations of farmers by chemical salesmen and the agricultural colleges whose research projects were often financed by these same chemical companies. Over the 20th century, the perception that agricultural soils were by and large a purely mineral medium and devoid of any worthwhile life became familiar and then comfortable. Along the way, a curious phenomenon began to develop. Crop pest infestations, fungi and viruses, previously unknown, overtook and destroyed formerly healthy fields. Simply maintaining previous yields required increasing amounts of these synthetic and organic fertilizers, and increasing amounts and new formulations of pesticides to combat plant infestations and diseases. While these synthetic chemical applications increased, erosion was accelerating. Soil failures broadened, and soon we found ourselves standing at the confluence of the Mississippi and Ohio rivers, watching as turbulent river waters hourly packed up hundreds of once rich and fertile farmland topsoil and shipped them off towards the Gulf of Mexico. The darker side of the Green Revolution was troubling. A while, a small remnant of soil microbiologists tenaciously clung to their beliefs that soil fertility sprang from soil life. For decades, they toiled away with much theory and little fruit. In the late 1980s and 90s, they suddenly gained access to high-powered microscopes that had previously only been available to medical researchers. These microscopes proved that much of what had previously been literally dumped into the laboratory trash can was a veritable gold mine of soil fertility and, even more wonderfully, of human nutrition. Previously unexplored magnifications brilliantly illuminated amazingly vast, dense, and complex soil ecosystems. The long elusive proof had been unearthed, and it came in unimaginable quantities that, like the ocean's depths, we are far from being anywhere close to having fully explored them today. Fertile, healthy soil is not a lifeless chemical medium occasionally frequented by enemy insects intent upon destroying our food supply. It's inhabited and even densely saturated by more life than we could ever have imagined. Trillions of soil life forms can easily exist in just half a teaspoon of healthy soil. We may have identified as little as 13% of soil's microbial organisms, and of those identified, many, if not most, are not well understood. Elementary students have long been taught that plants can make all the food they need with only sunlight and water. False. This is entirely false. In ancient times, plants are believed to have been entirely dependent upon a form of fungi known as mycorrhizae for sustenance. While many crops today generally survive either without or with minimal mycorrhizae, they still thrive best when living in community with them. The mycorrhizae and host plant relationship is a bit like a marriage. Like a good husband, mycorrhizae diligently provide for and protect the plant, and the plant likewise responds like a wife by feeding the mycorrhizae with carbs it cooks up, and lots of them. Mycorrhizae grow as microscopic fibers out of the plant's root and root hairs, branching out like veins into nutrient superhighways that crisscross and create a vast nutrient transportation network. In the process, they anchor the soil in place like a woven mat and extend the root system's reach far beyond the plant's own abilities. Individually 1/25th the diameter of a human hair, mycorrhizae fungus fibers are normally invisible to the human eye. Their size allows them to delve into rockier soil areas, otherwise unnavigable, to exponentially larger root hairs and roots. More than a mile of a single strand could fit inside a thimble. The creeping process of rock weathering by wind and rain was formerly believed to break minerals down into small enough particles for plants to absorb. Today we know that isn't true. Plants cannot convert inorganic soil minerals and nutrients into usable forms, and they are limited to what is immediately available in the soil around them, unless mycorrhizae help them. All along its strands, mycorrhizae exude powerful carbon-rich enzymes into the soil. These enzymes saturate nearby inorganic minerals and nutrients, slowly drenching and dissolving otherwise bound and inaccessible soil minerals and nutrients into forms that plants can absorb and digest. The fungi then brings home the bacon and transports these now digestible mineral forms back to their plant partner. Mycorrhizae-supported plants contain significantly higher amounts of vital nutrients such as zinc, iron, calcium, magnesium, manganese, and phosphorus than those without. They're naturally higher in nitrogen also, an essential building block for all protein. Should a harmful nematode or enemy worm be headed in the direction of its plant, the mycorrhizae will flex the tip ends of their fibers into a loop, creating a lasso or snare in the path of the approaching nematode. As the unsuspecting nematode worms its way through the loop, the snare suddenly tightens, lassoing the nematode and holding it snugly captive. The mycorrhizae then sends out digestive enzymes that first dissolve and then devour the nematode from the inside out. Adhesive enzymes are another tool in the mycorrhizal arsenal. Harmful nematodes must only brush up against the mycorrhizae for it to release those enzymes. Once contact is made, the nematode cannot break away from these incredible adhesives. Powerful digestive enzymes will then pass through the point of contact with the nematode and begin to digest it from the inside out. These pre-digested nutrients are then sent back as ready-made, protein-rich food sources to the host plant. Without the mycorrhizae to protect its spouse, the harmful nematode would likely do serious damage to the plant. But when paired with the mycorrhizae, the nematode becomes an essential complex protein food source for the plant and its offspring. Another top priority for the mycorrhizae is constructing a healthy barrier between their host plant and plant predators within the soil. It does so by building a hard protective coating over the outer root cells, besides defending its host plant against harmful denizens of the soil. Mycorrhizae produces proteins that protect its host plant from harmful genetic changes and damaging heavy metals within the soil. Mycorrhizae also defend their host plant against a variety of fungal root diseases by producing their own special antibiotics that guard the host plant from infection. While the mycorrhizae husband is protecting and providing for the plant is busy taking those groceries and using them along with the carbohydrates they synthesize from the air and sun to cook up a nutrient-dense meal for the mycorrhizae. After the mycorrhizae digest the complex carbs, they exude needed carbon reserves back into the surrounding soil in the form of phytolipids or fats, and an amazing complex organic protein known as glomalin. Like gazillions of microscopic grain silos below the soil, glomalin also serves as a storage mechanism for perhaps as much as 1/3 of all the world's carbon. These stored carbon reserves can last for years until plants need them. Glomalin also loosens up hardened, compacted soils and creates vital oxygen pathways for our subterranean cities to breathe more easily. Far from a lifeless mineral medium, healthy living soil breathes. For many decades, we've known that our forests and oceans breathed, but the collective inhaling and exhaling of healthy, fertile soil microbes has largely been ignored, along with the infrastructure that makes it possible. As these microscopic soil civilizations exhale carbon dioxide, plants receive it, breathing in deeply. Plants reciprocally exhale oxygen, and the soil microbiome inhales it gratefully. Each has a distinct role, each needs the other. The beauty of this relationship is particularly evident under drought stress. When stressed due to drought or extreme temperatures, plants will willingly sacrifice their own growth to feed more carbohydrates to the mycorrhizae in an effort to strengthen the mycorrhizae. They trust the mycorrhizae to retrieve more soil minerals and nutrients for the starving plant that will help it through stressful times. This beautiful partnership also ensures that host plants are less likely to experience drought or stress in the first place. When rainfall is adequate, mycorrhizae store water and slowly release it to their host plant, acting as a vast multitude of tiny aquifers conserving and supplying water according to their host's needs. Of many yetis up and down the roots, these water stores help shelter plants during extreme heat or cold by serving as insulation, which respectively cool and warm root systems. All of this gives plants colonized with mycorrhizae a competitive edge over plants without mycorrhizae. The mycorrhizae provides its host plant with more nutrients than the lonely unwed weed can secure, resulting in faster growth and monopolization of nutrient, light, and water resources. This is why wilderness areas visibly withstand drought far better and longer than neighboring cultivated fields with fewer mycorrhizal resources. Amazingly, mycorrhizae are non-monophyletic, meaning they have not evolved from a common genetic ancestor. How do we classify and organize a species that doesn't genetically resemble the family we've put it in? In this case, we group and classify them by how they reproduce. Many experts believe that up to 90% of all plant species are designed to host some form of mycorrhizae and did when the earth was new. This includes crops. In 2007, around 150 species of mycorrhizae were known. Today we're at over 342 and climbing. For these reasons, researchers anticipate that the 342 currently known species are a mere tip of the iceberg. These nutritional rock stars are just one type of soil fungi, and most soil fungi are still unknown. One study over about 3.5 acres of Swiss farmland recorded 2,554 types of fungi. Almost 2,000 of these we know too little about to classify them beyond simply being fungi. The same 2014 study, using these same samples, measured 3,877 types of soil bacteria units. Of these, scientists could only positively identify 61 species. Each has a specific function and a specific purpose. This was beautifully illustrated by a 2017 study in which scientists sought to identify the types of bacteria and fungi that grew inside of wheat plants. These are beneficial native bacteria and fungi that are essential to plant growth and live inside the plant. 18 unique fungal communities and 20 bacterial communities grew within these wheat plants from seedling to harvest. Like a symphony, different bacteria and fungi emerged and catalyzed plant growth at each growth stage. Within these communities, over 31 million distinct bacterial and fungal RNA sequences were counted. The majority of these could not be identified any further than that they were fungal or bacterial and previously unknown. Pure sunlight and water alone weren't causing these seeds to germinate. It was an enormous and diverse symphony of fungal and bacterial communities that supported and perhaps even informed the seed, then the seedling, and lastly, the mature plant of when the proper time was for the next stage of life. Yet another study found up to 6,000 unique bacterial genomes have been found in just 1 gram of soil. Still another 2014 study comparing soil bacteria in Iowan cornfields with Iowan prairies found 5.1 million bacterial DNA sequences in soil samples. To quote the study, the volume of genetic data recovered from these soil bacterial genomes was so immense that a single metagenomic project can readily generate as much or more data than is in the global reference database. Only a handful of these studies have been carried out, but it will take some time to glean through them. Soil bacteria themselves, for the little we know about them, are quite amazing. If you've ever caught the beautiful earthy aroma of freshly tilled, rich, healthy soil, then you've smelled the bacteria, actinomycetes. For decades, researchers struggled to classify actinomycetes, which had characteristics of both bacteria and fungi. The dark brown and black pigments often associated with healthy soils are also produced by actinomycetes. Probably the most important bacteria you've never heard of, this family of soil bacteria have played and continue to play a vital role in human history for their intelligent anti-cancer, antibacterial, antifungal, antiviral, and enzymatic properties. Popular antibiotics such as neomycin, tetracycline, streptomycin, and actinomycin all originated in soil bacteria. So did ivermectin. Today, over 5,000 compounds, up to 90% of commercially available antibiotics, and more than 70 individual antibiotics can hail some part of their origins back to these highly complex soil microbes. Like mycorrhizae, some actinomycetes species will inhabit a host plant and then produce natural plant protective antibiotics from inside of the plant to defend it and likely those who consume it from infection. We still don't entirely understand why these naturally occurring antibiotics are in our soil. They are the most genetically complex bacteria on the planet. Streptomyces colicolor, for example, has a genetic code with 8,667,507 base chromosomes, the largest number of genes ever discovered in a bacterium, any bacterium. Within these genes are more than 20 clusters that govern unknown or secondary metabolites, and an unprecedented proportion of regulatory genes. Researchers hypothesize that most of these regulatory genes are likely to be involved in responses to external stimuli and stresses. In other words, these millions of genes inside of the bacteria likely serve to guard and protect their plants. Evidence suggests that they produce plant growth promotants and inhibit harmful microbes and diseases better than man-made fungicides. As human antibiotic resistance grows, researchers are heading to increasingly remote areas to search for new actinomycetes species. Man-eating Bengal tigers, king cobras, saltwater crocodiles, pirates, and an endless labyrinth of gray clay estuarian channels shaded by thick mangrove jungle doesn't sound like the ideal soil microbiology lab, but it's in the Sundarbans of eastern India and other untouched soils that medical researchers are searching for previously undiscovered species of actinomycetes. Pharmacological companies are sending teams across eastern India, Bangladesh, Indonesia, the forested slopes of the Mongolian steppe, and even near the Temple Mount in Jerusalem. Why does this incredible abundance and diversity of soil antibiotics exist in the first place is an almost baffling question. Like many bacteria, actinomycetes certainly play a vital role in decomposing decaying plants and manures and converting atmospheric nitrogen into usable plant forms. Could they also be inherently healing and protective of us, the plant consumers? Another of soil's essential citizens is algae, and like much of the soil microbiome, we know little about it other than its existence. Because soil algae growth relies on sunlight, it only grows on the soil's surface, but its work is powerful in helping soil particles stick together. This creates natural soil agglomerates that open air spaces and increase airflow for the soil microbiome below to breathe. Algae nourishes the soil by pulling in atmospheric nitrogen and building it into protein forms the soil microbiome can potentially use. What's especially fascinating is that soil algae seems to flourish even in desert soils with sparse soil microbes. Dead plants, manures, and animals. The end of life above the soil marks the beginning of soil's life cycle below it. Decaying organic matter attracts ever insatiable soil scavengers, bacteria, fungi, and microbes. Shortly afterwards, these scavengers expel these nutrients. They are reconstituted in the form of complex organic molecules, collectively known as humic substances or soil acids. In the soil microbiome, nothing is wasted. These humic substances are nutritionally dense soil pantries that provide healthy meals for both bacterial and fungal grazers, the herbivores and omnivores of the soil. An excellent marker of soil health, soil acids provide a benchmark of available food resources for the soil microbiome. As such, they are foundational to topsoil's creation, sustenance, and rejuvenation. Healthy topsoils produce soil acids continuously, but because the process is bacterial-driven, they do it so fastest in warmer climates and weather. In Antarctica, humic and fulvic acid production slows to a near halt. In tropical climates along the equator, soil acid production can occur swiftly. Like most of life, though, the healthiest soil civilizations tend to thrive best within moderate temperatures rather than extremes. As scientists are just beginning to understand soil acid production, they've been faced with a challenge of classifying and naming these amazing substances that don't fit into any previously recognized categories. Soil acids aren't animal, vegetable, or mineral. They are carbon, oxygen, and hydrogen clustered together with a wide variety of other elements to create enormous groups of DNA, amino acids, and carbohydrates of immense size. The size of a soil acid when compared with most previously known chemicals has been likened to comparing a salmon to a whale. Poly just means many, and that's what these soil acids are believed to be. Poly what is the question, though? Like snowflakes, their individual molecular structures are unique, even from their nearest neighbors, and vary greatly in both complexity and diversity. This diversity is essential to healthy living's topsoil, and yet they are simply too diverse and too complex to fit soundly in any existing chemical or biological category. For the time being, we call them soil acids. We can group them together by color, by temperature preferences, and by carbon content. But everyone knows that the term soil acids is just a vague colloquialism we're using as a placeholder for lack of a better understanding. Adding to the complexity, many of the bacteria, fungi, protists, and nematodes that created these soil acids haven't been identified yet. In wine, cheese, or preserved meat production, different types of bacteria and nutrient levels allow for nearly limitless varieties of scrumptious flavors, enticing shades, and appetizing aromas. Soil acid production is no different. The amount and diversity of nutrients provided by decomposing organic matter combined with the quantity and diversity of the soil microbial species determine the amount, types of nutrients, and even the color soil acids lend to their home soils. Coppery gold soils may be rich in fulvic acids, while humic acids tend to turn the soil a rich dark brown. Soil acids are the soil microbiome's first choice buffet. They also, like mycorrhizae, will over time slowly dissolve the minerals and rocks surrounding into plant absorbable forms. In times of abundant rainfall, soil acids absorb water into their structures, transforming and expanding from solid to gelatinous substances that can hold up to 98% of their weight in water. Like mycorrhizae, they too become widespread mini aquifers that in drought or extreme temperatures provides plants with both an additional reservoir of water and a natural means of cooling or heating root systems. Under heavy rains, water-saturated soil acids can link together with other soil particles to form an invisible water-resistant fabric on the soil surface, which helps to resist erosion from damaging winds and rains. When combined with plant cover and mycorrhizae netting, soil acids, the raincoat of the soil, provide serious defense against soil erosion. During their short lives, soil microbes have the potential to reproduce and multiply with incredible speed. To an extent, this is a blessing. When soil fungi and bacteria die, their bodies release stored organic nitrogen in the form of complex proteins and amino acids in addition to other nutrients back into the soil, further feeding the soil microbiome and the plants it partners with. Were it not for soil predators, though, soil bacteria and fungi might completely devour the world in short order. Thankfully, have checks and balances in the form of soil protists and nematodes, the bacteriivores and natural predators of the soil. No one really knows what a protist is, and that's part of their charm. Invisible without extreme magnification, these peculiar living creatures called protists are neither animal, nor plant, nor fungus. Despite our laboratory advances in preserving soil microbes, protists continue to be a challenge to extract from farm soils. They are delicate, ornate, and jewel-like, yet very strong. Like mycorrhizae, they're mostly non-monophyletic and did not genetically evolve from a common ancestor, once more complicating our studies of them. Unlike mycorrhizae, they're classified by how they move rather than reproduce. Generally single-celled, they ooze or flutter, depending upon their constitution, through the soil, riding upon seeds of water molecules to wage war upon other protists and feast on bacteria. One such group of individuals is the naked amoeba, whose gelatinous blob of a body, bizarrely, can, sans muscles, senses, or nervous system, chase down, engulf, and consume its prey. Some refer to this as a hunting style, others calling it a grazing aptitude. But regardless of whether you see them more as a lion or more lamb-like, their role is essential to a healthy, functional soil microbiome. Biofilms are sticky, slimy substances that some bacteria produce and shield themselves with as a protective defense mechanism. Recently, they have come to the forefront of human medicine as potential root causes for a variety of bacterial diseases. Some protists seem to have been specifically designed with equally protective exteriors that allow them to wade unharmed into those biofilms and graze on these bacteria. Like velociraptors or birds of prey, raptorial protists claw, grasp, and tear apart bacteria and other protozoa as they devour them. Heliozoan protists look, as their name implies, like mini suns that wrap up their bacterial prey in happy sunbeam-like extensions before absorbing them. Nematodes are microscopic worms. A couple of billion of them could easily fit into a teacup. But you won't find anywhere near that amount in healthy topsoil. Within a square yard of healthy farmland topsoil, you'll likely only find a couple million. Like soil bacteria and fungi, nematodes are another tremendously diverse group. There are probably as a million distinct species of nematodes. Contrast this with a little more than 5,000 unique species of mammals in existence, or perhaps the 10,000 species of reptiles, for a sense of perspective. Until recently, nematodes were synonymous with massive crop root destruction, and the only good nematode in decades past was considered a dead one. Yet many, if not most, nematodes don't eat plants. They eat soil bacteria and fungi, and lots of it. Some are even equipped with a straw-like mechanism that can slurp up bacteria 20 or more at a time. A 2017 study of the German wheat farm soils found the nematodes in their soil samples fell into 5 distinct groups: plant eaters, bacterial eaters, fungal eaters, predators, and omnivores. A little more than half of the nematodes in their samples ate primarily soil bacteria and fungi. A little less than half dined exclusively upon plant roots. And unsurprisingly, very few, only 1.3%, were either omnivores or predators. Even in healthy above-ground ecosystems such as prairies or forests, predators comprise a small percentage of the total number of creatures. Too many predators within the soil would decimate prey species. Too few predators and prey species would overrun and destroy available vegetation, leading to the prey species' ultimate starvation. Nematodes are essential to healthy soil and their soil microbiome balance. As we peer into the microscope, a miniature subterranean ecosystem that mirrors those seen in our forests and prairies sharpens into focus. Every species, even predatory species, are absolutely essential. In 1926, the last wolf of Yellowstone National Park was killed, and the species was considered to have been completely eradicated. From a pioneer mindset, it made sense to prioritize our own survival and that of a key food source by eliminating what was considered to be a dangerous predator. But there were other unexpected consequences. The park began eroding down the river. Park riverbanks were literally washing away. Rangers thought that they were preserving the elk herds by eliminating the wolves, but pressure from the wolves had kept the elk moving along and from grazing any one area too intensively. Without the wolves, the elk overgrazed young willow and aspen trees, whose roots and root partners had helped to hold the soil in place along the riverbanks. For the next 75 years, the cascading damage continued, and even ultimately shifted the course of the park's rivers. It became apparent that an entire ecosystem was dying, and all because of the removal of just one species. Despite protests and fears that they would devour all of the park's elk, a small pack of just 14 Canadian wolves were introduced to the park in 1995. When the wolves returned, so did the former bounteous ecosystem. Willow and aspen trees grew again, the songbirds and beavers returned. Riverbank soils were stabilized. Balance is essential. In addition to the carbohydrates and fatty acids their plant wife cooks up and feeds them, mycorrhizae hunt down and consume nematodes. Without nematodes, mycorrhizae lose key complex protein and fatty acid components of their diet that they can no longer share with their plant wife. The plant wife, in turn, can no longer provide these complex proteins and fatty acids to animal or human consumers. Like wolves, bears, sharks, and other predators that were formerly demonized, but now have been recognized as essential to ecological health, nematodes are starting to be recognized for their critical role in soil health. Whether above or below ground, predator species keep herbivore species and vegetation in balance, and soil bacteriivores, ultimately strengthen soil bacterial populations by picking off the weak and the stragglers. Balance is as highly prized within the soil microbiome as it is in forests or prairies. Decimation of any one species creates imbalance within the entire soil food web. Every community member is essential. Predators and prey, omnivores, herbivores, and carnivores, regardless of how inconsequential one of these minuscule individuals seems, the collective health of the entire subterranean microbiome depends upon these individuals doing their jobs. To provide a diverse and a rich array of nutrient resources for themselves, for their plant wives, and for plant consumers, each of these communities must be present and in balance with one another. It is these orderly and balanced communities whose industrious citizens build and maintain stable soil infrastructure, a defense system, and a food and water supply that benefits all communities. In a thriving soil microbiome, its tiny citizens are ever growing, producing, reproducing, being eaten alive or dying, and ultimately are recycled back into the soil microbiome at an incredible speed. A crescendo of complex nutrition is fabricated together as nutrients are developed and transferred from decomposing materials to bacteria, to nematode and protist, to mycorrhizae, to plant consumer. Nothing is wasted. Even when soil microbial bodies die unconsumed, their bodies disintegrate into organic proteins, complex carbons, and fatty acids for other members of the soil's food chain to consume. Each individual within the greater collective community plays a vital role in the soil microbiome, and without one of these elements, soil's ability to nourish plants and ultimately people is diminished. When too much unchecked power or advantage is given to one population or another, a soil microbiome out of balance quickly becomes a fierce, tribal wasteland. Soil science once seemed a bit like geology, where we could sort soil like rocks by their colors and textures. We could measure soil particle size and texture. We could weigh soils. We could carefully procure soil samples, put them in a petri dish, and examine them under a microscope. We could only see a barren wasteland. It turns out we simply weren't looking closely enough. Now we see that soil science is more akin to marine biology than geology. Soil microbial species are simply too dynamic and too sensitive to be observed by yesteryear's laboratory and research techniques. Farm soils can never again be seen as a mere mineral medium and chemical conduit. We've come full circle to the agriculturalists our forefathers were, who intrinsically knew that fertile soils were teeming with rich, abundant, and healing life, even if they couldn't prove it. It is that soil life that is essential both to the health of the plants grown within these soils and the animals and people who consume them. When we return to the desolate shores of Leptis Magna, when we see the sands of the Sahara strewn about in the atmosphere above U.S. farm soils, when we stand on the banks of the Mississippi and watch the turbid waters carrying American farmland off to the Gulf of Mexico, the question is obvious. If the soil microbiome is the soil's inherent natural defense against erosion, why isn't it doing its job? This is the end of Chapter 2. Thank you so much for listening. Please like and subscribe if you'd enjoyed this, and you can receive notifications when the next chapter comes out. I'd also love if you go over to my Patreon page. The link is in the notes below. And you can subscribe there as well for more in-depth, behind-the-scenes details about some of the information in each chapter. Thanks so much. Bye-bye.